interferon alpha in the form of innate immunity. Now, interferons, because they have evolved for a number of years along with viruses, and because they are the first line of defense, somehow the mechanisms of interferon alpha also get intertwined with the generation of adaptive immunity, so that if a patient is given or produces interferon alpha along with his or her infection, the adaptive immune response also gets helped. Antigen presentation is better, right kind of cytokines are produced, and your long-lasting immunity takes place. Next slide, please. What is interesting, though, is that while uh, most respiratory viruses induce interferon alphas or type 1 interferons, interestingly, COVID-19 does not do a good job of doing that. So this important paper came out in Cell in the year 2020 from Blanco Malo's group. And what it shows is a comparison of sera samples of 24 COVID-19 patients with those of 24 negative controls, and compared. If you look at the left panel, interferon beta is circled, that COVID-19 patients actually did not produce any type 1 interferon alpha. Specifically tested here is interferon beta, so we can talk about interferon beta only. But this is indicative of the fact that even though there has been a viral infection, an RNA virus, a respiratory infection, and therefore a type 1 interferons alpha, type 1 interferons should have been produced, what we're seeing is actually the levels are undetectable and like that of negative controls. However, that does not mean that the body did not recognize this as a threat or ignored it, because if you look now on the right side of the slide and look at some of the inflammatory cytokines such as IL-6, IL-1RA, and chemokines, so many of them shown there, you find that in almost every single case, there has been an increased production of inflammatory cytokines and chemokines, showing that the body is looking at it as an inflammatory threat, but still not doing anything about the virus itself. Next slide, please. Sorry. So this paper came out from the group of Hadja et al., and they looked at whether the type 1 interferon alpha responses were impaired with respect to disease status. So if you look at panel B, we are looking at interferon stimulated genes. So as interferon alpha is produced by one infected cell and gets secreted from that cell and goes to neighboring cells, binds to the receptor, a JAK-STAT pathway ensues, and activating a number of genes by a group, they are called interferon stimulated genes. And you find that if we still focus on panel B, the first bar, black bar, is healthy control. So obviously you do not expect ISG score to be high there, interferon stimulated gene score to be high there. But when you look at mild to moderate disease to severe disease to critical disease, actually the ISG score surprisingly seems to go down. You would have wanted it to remain high, but you find that patients who became moderate and severe actually had poor ISG scores. If you actually look at levels of interferon alpha produced themselves, again the same pattern, panel C, as disease progresses, as you look at patients who have progressed into severe disease or moderate disease, they have lesser interferon alpha production compared to the milder patients. And all of these scales, at least the panel C is a log scale, so those differences are very meaningful. Still, another important piece of information came out of the group of Paul Bastard et al. And what they looked at was a number of severe disease patients, 987 patients with life-threatening COVID-19 pneumonia, 663 patients with asymptomatic or mild disease, and also a large group, 1,227 healthy controls. They took their sera sample and tested for the presence of autoreactive antibodies, especially neutralizing antibodies against type 1 interferon, type 1 interferons. This slide, if you look at the bottom panel now, on to the left, uh, the y-axis is fluorescence intensity, which is representative of uh, amount of autoreactive antibodies present in the patient. Uh, the higher the value, the higher the amount of autoreactive antibodies. If you look at those patients that had life-threatening COVID-19 disease, many of them had autoreactive antibodies to not only interferon alpha, the red dots, but also interferon omega, blue dots, showing that in general, autoreactive antibodies existed in those patients who progressed to life-threatening COVID disease. Not all of them, but many of them. And of course, asymptomatic individuals did not have autoreactive antibodies. Healthy controls also did not have as much as the advanced life-threatening patients had. These antibodies were, so sera samples were tested and found to be neutralizing in nature, so that when you try to infect cells with a virus, COVID-19 virus, and then do that in the presence of varying amounts of the plasma from these severe COVID disease patients, you find that ISG scores or induction of STAT1 in this case seems to go down dramatically as the serum concentration increases. So it shows a correlation again in another way, that there are patients who either do not express interferon alpha sufficiently, type 1 interferon sufficiently, or unfortunately happen to have autoreactive antibodies to type 1 interferons, and all of those two types of patients are the ones actually that progress to more moderate and severe disease. These authors actually went back to the freezer and looked at the first sample also, uh, which was taken at the time of entry of patient into the hospital, and found that those patients who had high antibodies against interferon alpha later in the disease actually were carrying these antibodies right from the beginning. We all know that antibody responses take around 2 weeks at least to show up, IgG antibody responses. So when a patient had just entered the hospital and was probably 7 days into the disease, there was no reason for him or her to have uh, antibodies. So these antibodies existed in those subjects naturally, and it was those unfortunate subjects that ended up becoming severe in this group of patients studied by Paul Bastard et al. Uh, Still another paper. Now, what is the impact of the clinical treatment of these patients with interferon alpha? So this paper came out of Wang et al.'s group and looked at retrospectively patients from Wuhan, 446 in all, out of which 242 patients happened to have been given type 1 interferons. Now, we all know that China was the first to suffer from COVID, so obviously they were the first to try many things, and type 1 interferons, interferon alpha 2b more specifically, was one of the drugs that they had tested. Interestingly, what they found was that in retrospective analysis, those patients who happened to get interferon alpha early in the therapy had reduced mortality and also showed better responses than those patients who were given lopinavir or ritonavir. Another paper coming out of the group of Lu et al. Happened to show an interesting phenomenon, which we will show, my colleague Dr. Kevin Consagra will show from our own clinical trial, a reproduction of findings of the study that came out of Lu et al. Now, Lu et al. Also did a retrospective analysis only of their patients and found that those patients who were on glucocorticoids and also happened to get interferon alpha therapy actually benefited the most in comparison to others. So this combination of glucocorticoids with interferon alpha therapy giving a benefit and resulting into earlier hospital discharge, earlier symptomatic relief, was postulated by these scientists in the following manner. Glucocorticoids, as we all know, are anti-inflammatory, and they are reported to actually suppress the type 1 interferon alpha production. And when you um, are giving patients glucocorticoids maybe earlier into the therapy than they should be, they would naturally stop producing any more interferon alpha. And when you supplement that interferon alpha also, you get uh, the antiviral effect along with the anti-inflammatory effect, resulting into better outcomes. Still another paper, Wang et al., comparing a smaller group of patients, 22 patients who were given lepinavir alone, and 19 who were given LPVR in combination with interferon alpha. And it was clear, if you compare red bar with the blue bar, and the y-axis is the length of hospitalization in terms of number of days, that those who got LPVR only had to stay in the hospital longer than those who got LPVR along with interferon alpha 2b. And within the interferon alpha 2b group, although the sample size was small, they could also dissect subjects into those who got early treatment with interferon alpha and those who got later treatment with interferon alpha. And it is beautifully clear, as any scientist would postulate, interferons, which are supposed to be the first line of defense, would work the most only when they are given early in the disease. Green bar shows that effect very strongly. I now bring you to our own in vitro data and then in vivo human data that Dr. Kevin Consagra will be presenting. But here you can see in the blue panel on the left, the IC50, EC50 values of antiviral effects of various molecules that have been tested uh, in viral infections, and a few of which have been tested in COVID also. So as you can see, remdesivir is more potent than favipiravir, and PEG interferon alpha is more potent than anybody out there, simply because you go from micromoles to femtomoles. So almost a million times, at least 100,000 times more potent PEG interferon alpha 2b is compared to remdesivir. And interestingly, the remdesivir IC50 value you are unable to reach in the blood with the recommended doses of remdesivir, because that would end up being toxic to the patient. Whereas PEG interferon alpha 2b, at that 0.3 nanogram per ml IC50, you are very easily able to achieve in human patients. So for example, if you now focus your attention to the orange box at the bottom, healthy volunteers which were given 1 microgram per kilogram dose ended up having a Cmax value of 0.65 nanogram per ml, which was already twice the amount of 0.3, more than twice the amount of 0.3 nanogram per ml, which was effective in terms of IC50. So with these bits of knowledge, some of which came out before we started the trial and some during our conduct of phase 2 and phase 3, we will now look at how our molecule ended up behaving in clinical trials. And I would request my colleague, Dr. Kevin Consagra, to take over from here. I will run the slides for you, Kevin. Just let me know when to move them. Thank you.
Thank you so much, Dr. Sanjeev, for your innovation and showing off of the science beyond the giving the interferon. So now I would like to request Dr. Kevin for your practical implementation of the interferon, pegylated interferon Virafin for COVID patient, please.
Thank you, Professor Coco. So, shall we go to the presentation of the clinical part?
Dr.
So I mean, whoever was displaying the slide deck. Can you please put the slide deck on?
Dr. Kevin is a general manager in clinical research and development of the Zydus Research Center Medical Leads clinical research project to development of NCE, NBE, and biosimilar. He has a many experience for using the Virafin. So we would like to request Dr. Kevin Kensakara for showing the us for the practical implementation of the Virafin. Dr. Kevin, please.
Yeah, thank you, Professor Ko. So yes, in the background, I think Dr. Talwar and Dr. Sanjeev has made my job more easier because they have given a proper background of the COVID-19 as well as the role of the interferon in the COVID. So in this background, before initiation of the phase 2 study, we have okay. Before initiation of the phase 2 study, we have a healthy volunteers data of the 1 microgram per kg dose. And at the time, in the last slide, you have seen the Cmax as well as the AUC comparison with the in vitro data. So we are matching with proper efficacy with the interferon alpha 2b. It's a pegylated one. And on that background, we have initiated the study, the phase 2. And in this phase 2 study, we have a dose of 1 microgram per kg. And we had also in the provision in the protocol to use a second dose, but there is no requirement of the second dose. In the study, this is a proof of concept study. So we have started with 40 participants, and the 20 participants has given the PEG-IFN and the standard of care, and another 20 has received only standard of care. In the enrollment point of view, we have just focused onto the moderate subjects, and our primary objective was, if you see the blue one, the third and the fourth point, the hospitalized and the intubation oxygen, those subjects are participating into the study. And we have put the improvement, and the subject is not hospitalized and there is no limitation of the activity. So there is a change into the 7-point ordinal scale of the WHO. Those will be considered as the primary objective. And in other secondary objective, we have a qualitative RT-PCR as well as the occurrence of the supplemental oxygen, mechanical ventilation, duration of the hospitalization, safety and the tolerability, the pathological, all the kind of the hematology, biochemistry markers are there, and as well as the biomarkers, because first time we are using this into the COVID subjects. So we have a thorough biomarker evaluation of the CRP, interleukin 6, D dimer, interferon gamma, ferritin, TNF alpha, as well as the interleukin 1 beta. And the different days, like on the baseline, day 3, day 7, day 10, as well as in day 14. We have a demographic, the male and female subjects were part of the study. And in this study, the subjects with the hypertension, diabetic mellitus, as well as the asthma subjects are there. Here, the sample size on lower side. So we have a small number of the comorbid condition is there. But in our phase 3 study, we have enough number of the subjects who have a comorbid condition and they were part of the study. So now if you see the primary objective, the improvement into the WHO ordinal scale, and if you see the yellow box, the test group, we have 20 subjects, and out of these 19 subjects as an improvement in the WHO ordinal scale, the percentage is 95%. And if you see the reference group, 13 subjects has improved out of 19 subjects, so that is 68.42. So that value is significant. So compared to the only standard of care group, if we are adding the PEG interferon alpha 2b in the standard of care, there is a definitely significant level is there in the WHO ordinal scale. And now afterwards, this particular product is an antiviral product. So we have different days for the evaluation of the RT-PCR. And if you see on day 7 on the test group, we have 80% of the subjects, those are negative with RT-PCR. And the reference group, that number is 63%. But now if you see on day 14, the 95% of the subjects are the RT-PCR negative, and the 68% of the subjects are the negative into the reference arm. So definitely on the test group, we have a higher number of the responders are there. And if you see the scatter plot, of the oxygen requirement. The red one is the pegylated plus the SOC, and this blue one that is only SOC. And if you check the scatter plot, they are onto the lower side. There is a less requirement of the oxygen compared to the only standard of care arm. And the similar kind of the results we have also produced in our phase 3 trial. And this is a small sample size, but in phase 3 we have a higher sample size and we have a similar kind of the findings are there. The biomarkers, because sometimes the KOLs are thinking maybe if you are giving the pegylated interferon, maybe there is a chances of the cytokine storm. So we have a thorough evaluation of all the biomarkers in this phase 2. And if you see on day 3, day 7, day 10, day 14, as well as day 29, there was no significant difference observed between the treatment groups of these biomarkers during the study if we compare with the standard of care arm. And if you put a conclusion on the basis of the safety point of view, the no clinically relevant any kind of the significant finding on the basis of the vital signs ECG, and the overall the single dose of the pegylated interferon alpha 2b was safe and well tolerated. Not a single subject has any of the adverse event that may be due to the interferon alpha 2b. And on that background, we have initiated the study with the large sample size of the 250 subjects and across the India. And in this study, we have total samples is 250, 1:1 randomization. And if you see the standard of care group, we have 125 subjects. And in the standard of care plus the pegylated interferon alpha 2b, the another 125 subjects are there. In this group, we have your dose is 1 microgram per kg. That is, we are using subcutaneously. And we have targeted the population of the subjects with the moderate COVID-19. And we have a housing till day 7. And it may change depends upon the clinical condition of the subjects and sometimes the regulatory guideline as well as the current hospital practice. And here we have a primary objective was 2-point improvement into the ordinal scale on day 11 plus or minus 1 day. And we have put this 7 scale as per the WHO. And the secondary objective, we have a different days like day 8, day 11 and day 15 for the improvement in the WHO ordinal scale, as well as the subjects who are the RT-PCR negative, the requirement of the supplemental oxygen, mechanical ventilation, duration of the hospitalization days. Here we have added the new endpoint like time to resolution of the clinical signs and symptoms, the incidence of the adverse event as well as the serious adverse event, and other kind of the change in the WBC count as well as the biomarkers. Inclusion criteria of the study, we have preferred the guideline of the family and the health and welfare of the India. And in that guideline, the moderate subjects they have defined, like the subjects is respiratory rate is more than or equal to 24 breaths per minute, or the SpO2 is in between 90 to 94%. Otherwise, the subject should be RT-PCR positive, as well as the radiographical infiltration is there. And exclusion criteria, we have excluded the subjects of the ALT and AST more than 5 times, the stage 4 kidney disease, pregnant and the breastfeeding female, the severe comorbidity like uncontrolled hypertension as well as the diabetic mellitus. In India, we have a recommendation to use of the hydroxychloroquine. So we had a baseline ECG to the subjects who have a higher QT interval. Those subjects were also excluded from the study. And this is a schedule of assessment. We have a screening window is 5 days. Then we have a check-in and the dose out, check-out is on day 7. And then we have a safety follow-up till day 29. Now, if you go to the result, before going to the result, if you see the subject disposition, so we have 120 subjects into the test arm and 130 subjects into the reference arm. Number of subjects discontinued from the study in the both arms remains similar. There was no any mortality that is due to the pegylated, and couple of subjects are also lost to follow-up as well as the withdrawal subjects by the study. But in this study, we have put a higher sample size considering the dropout. So we have achieved a good power of the study to evaluate the primary objective as well as the secondary objective. As I have mentioned earlier, we have a higher number of the subjects with the comorbidity, like the type 2 diabetic mellitus, we have 59, and the hypertension subjects are also 58. And if you see the demographic characteristic of the age, gender, weight, height, and the BMI, we have an equal distribution of the subjects in both the groups. And if you see the p-value, that is non-significant. So the equal distribution of the subjects are there in the test as well as onto the reference group. And the common treatment that was prescribed during the study, that was hydroxychloroquine, antibiotics, steroids, vitamins, anticoagulants, and the antiviral therapy, that is as per the investigator discretion. Because in this phase 3 trial, we in India, we got a approval of the remdesivir for the emergency use. So some of the investigator prefer to use the remdesivir and some of the investigator do not prefer to have a remdesivir. And the favipiravir is not given to the subject. So in our analysis, first we have analyzed as per the protocol, all the subjects together, and then we have a subgroup of the population. They have received the steroid, and some group of the population they have also received the remdesivir. So we also did a subgroup analysis of the subjects, how the pegylated will behave with and without steroid as well as with and without remdesivir. So those slides I have also captured in this presentation. And first, as we know, we have a viral phase in the initial COVID-19. So on day 11, if you see the 2-point improvement, so there is no difference between the test and reference. So within day 11, all the subjects are improved. So around 91% in test arm and the 92% into the reference arm. But the theory of the antiviral therapy is early the therapy, that is the better. So this particular pegylated alpha 2b, we have already put a POC study. The drug has an antiviral activity and as well as the 2-point improvement if you are giving the pegylated early, as well as the 2-point improvement improved on the day 8. And if you see on the day 8, the value of the test arm, the 80% subjects are the responders here, and the 68% subjects are responder into the reference arm. So that value is significant. And then we go to the, this is a bar diagram, those similar values are there. And now if you go to the RT-PCR, as I have mentioned earlier, the antiviral activity of the pegylated, the early the therapy, the early the RT-PCR negative. So on day 7, if you see, the 91% of the subjects are negative with the test arm, and the 78% of the subjects are negative onto the reference arm. So the p-value is highly significant. The value is 0.01. And then we also did analysis of the supplemental oxygen. And if you see the duration of the supplemental oxygen, the 56-hour requirement into the test arm versus the 84 hours requirement into the reference arm. And the signs and symptom, the 5 days are there into the test arm and the 6 days are there into the reference arm. And the safety perspective, the drug is safe. There was not a particular adverse event that is related to the pegylated, and we cannot see any of the adverse event in the system organ class. The higher number of adverse events are there into the test arm, and the lower adverse events are there into the reference arm. So the adverse events are equally distributed in both the arms. So there was no any safety concern in this all the subjects. And as I mentioned earlier, we have additional analysis. So here, the first table that I have already narrated, all the subjects together, we have a highly significant value on day 8. But now then the left one table on the lower side, we have divided into the subjects who have received the remdesivir, and the right one, the subjects who have not received the remdesivir. The subjects without the remdesivir, the value is highly significant, like 0.006. 86% versus 68%. So yes, if some of the investigator they are not prefer to use the remdesivir, still the pegylated has a good role for the 7-point improvement into WHO scale. And similar way, day 11 and day 15, there is no change between test and reference part. And if you consider the SOC plus remdesivir, there is no change on day 11, day 15, as well as on day 15. And now we have a value of the negative RT-PCR. Now, if you check without remdesivir, the 93% of the subject has RT-PCR negative on day 7, and the 76% of the subjects who are the RT-PCR negative with the only SOC arm without remdesivir. So the value is also highly significant. So without remdesivir, yes, the pega-IFN has an antiviral activity. And if you see the RT-PCR result with remdesivir SOC, although the sample size is on lower side, like 28 and 27, so the value is non-significant on day 7, day 11, as well as on day 15. And then, as Sanjeev sir has mentioned, the role of the steroid in the COVID-19, and if you are adding the PEG-IFN with steroid, what will happen to the endpoints? So here, the first table, we have all the subjects together, and the lower one, the left side, if you see the steroids, like the PEG-IFN plus standard of care, we have used the steroid in 71 subjects, and the reference group, we have 74 subjects. The 85% of the subjects we have an improvement onto the 2-point ordinal scale, as well as in the reference group there are improvement in the 67% of subjects. So the value is highly significant, 0.009. And without steroid, here there is no change between the test and the reference arm. Maybe that is due to the less number of the sample size. But if you are using with steroid, yes, there is a good improvement into the subjects. And a similar way, we have RT-PCR value on day 7. So there is no change into the antiviral activity of the PEGA-FN if you are using with steroid. So 95% of subjects improved into the test arm, and the 73% of subjects improved into the reference arm. The value is 0.0002. So that value is highly significant. And the duration of the supplemental oxygen without remdesivir, yes, the value is significant, like 56 hours versus 84 hours. The p value is less than 0.001. Then we have subjects who have a standard of care with remdesivir. There was no change into the test and the reference arm, although the sample size is only 4 and 7. And the SOC without steroid, the supplemental oxygen, there is no change between the test and the reference arm. And if you see with steroid, the 56-hour, the duration of the supplemental oxygen required with the test arm, and the 84 hours with the reference arm, the value is significant with the test arm. And then we have a clinical signs and symptoms. So without remdesivir, the 87 subjects are there into the test arm, and 89 subjects are there into the reference arm. But if you see the median range, 5 days versus 6 days, so there is definitely a significant improvement into the test arm. And without with remdesivir, there is no change. And if you see SOC without steroid, the sample size is 44 and 42. The median improvement of the signs and symptoms remain 5 days in both the groups. And if you see SOC with steroid, the median value remains 5 days for the test arm and the 6 days for the reference arm. So that is highly significant. And considering the data, as we have discussed earlier, this product is already available in India since 2011, and we have more than 2,900 patient years data. They are very safe and efficacious in the chronic hepatitis B and C infection. And if you see the result of the COVID-19, the higher proportion of the patients in the PEG-IFN were showing 2-point improvement on day 8. The p-value is 0.03. The RT-PCR negative by day 7, the higher proportion of the patients are there with the PEGA-FN arm. The p-value is 0.01. The hours of the supplemental oxygen required was significantly less in the PEGA-FN compared to the reference arm. The p-value is less than 0.0001. Resolution of the signs and symptoms are also there. The 5 days is there in the PEGA-FN and 6 days are there into the reference arm. So that is also significant. There was no any cytokine storm. There was no clinically significant changes into the laboratory markers. So on that basis, this particular product, the pegylated alpha 2b, is safe and very well tolerated. And we have got an additional indication, the treatment of the moderate COVID-19 infection in the adults from our DCGI. In the initial part, we have also tried to take an indication to the mild subjects. But as you know, the subjects, the SpO2 level is changing. It's not remain onto the similar way, because within 1 hour or 2 hour, so maybe the subjects are into the mild to moderate into the category. And the subjects with the moderate category, we have a very good improvement in the RT-PCR, in the 2-point WHO ordinal scale, the requirement of the supplemental oxygen, resolution of the signs and symptom, and there was no any safety concern. So on that basis, I just conclude my talk onto the clinical perspective. So thank you.
Okay. Thank you, Dr. Kevin, because your findings are very, very, interesting for us. So you means, according to your finding, paraffin can be given in the moderate patients at home because there is a single short injection subcutaneously. So it will be a lot better for the all the healthcare provider who are treating patient at home, because there is a single injection and the only SC injection is no need for intravenous therapy. So that will be very good. So thank you so much and for your all the experiment finding. And because of experiment finding, we would like to hear from the real world evidence. I mean, the real world experience by the data who are using the paraffin. So in that case, I would like to request and invite Dr. Nitin Agrawal. He is a consultant physician, diabetologist, and cardiologist, and managing director Ayush Hospital, Pajava, India. Dr. Nitin, thank you for joining us, and we would like to hear your experience about the using the paraffin for us. Thank you, Dr. Finn, please.
Yes. Uh, Good afternoon, everyone. I hope I'm audible.
Yes, sir.
Yes, very much.
Yeah,
So,
you're audible, sir.
so I'll be sharing some of my slides. Since 3 months, I'm using Virafin since the launch of Virafin in Karnataka, and I'm practicing at Bijapur, Karnataka district place, and we have treated around 25 cases, so which I'll be putting in just few slides. And absolutely, as Dr. Kevin has already elaborated about the interferon, pegylated interferon alpha 2b, we can see that at our place in Bijapur, uh, we have a 60 bedded hospital, and during the first wave of COVID, we treated almost around 1,000 patients of COVID. And my slides are visible, I guess. So during the second wave, we also treated around
See your slide.
600 patients. And we all know during the second wave, because of the change in the strain, we saw more mortality, more severe cases, and the same treatment protocol did not work in many of the cases compared to first wave. But we are very happy with the experience of using Virafin recently. And the inclusion criteria at our center, we used Virafin in 25 patients with pneumonia, diagnosed confirmed COVID-19, and all the patients were adults more than 18 years of age. They were all confirmed RT-PCR positive cases, and the presentation or onset of symptoms was less than 10 days. We selected, and their
Dr. Nitin,
cities, yeah,
Dr. Nitin, we cannot see your slides. We cannot see your slides. You have
okay.
slides?
Yeah, I'm presenting with a slide actually.
No,
I'm
actually,
already
we are not
sharing
able to
the.
see. You
Okay.
can share, use the screen and go to the slide.
Yeah, I did that. I'm using the screen.
Okay.
Just,
We
just
are not
one.
able to see it.
Yeah.
You can try again. Yeah.
So it's very easy, simple things. Whatever I am giving my experience, the slides also are showing same thing. So can I just
Sure.
put this orally?
Yeah, sure.
So out of these 25 patients, all were adults whom we treated at our center. The CT score was around 6 to 16, and their presentation, oxygen saturation, they all had hypoxia, mild, around 90 to 94% of oxygen saturation they had, and all were confirmed RT-PCR positive cases. We used in 15 moderate and 10 mild cases, and those having comorbidities, there were around 16 patients who were having comorbidities. They had diabetes, hypertension, and one patient had a cardiac ailment. And if you see the outcome part, almost 24 patients improved, except that cardiac patient whom probably we selected because he had multiple cardiac ailments, like he also had IHD and RHD both, and he had undergone CABG. Even though ejection fraction was good, but he died because of his cardiac ailment. And among these 25 patients, 17 were inpatients and 8 were treated on OPD or daycare basis. That is a plus point what I would like to highlight. So absolutely, even though they had mild hypoxia, CT score was more than 6. We took a chance and gave injection, and we monitored for 4 hours in our daycare center for the vitals or any side effects. But they did well. These 8 patients were treated on daycare basis with 4 hours observation, and they hardly had, out of 25, 5 patients had mild headache. Apart from that, we did not find any other serious side effects. Only 1 or 2 patients had some nausea. And for the headache, we just gave good hydration, and the headache relieved for these 5 patients. And the patient's blood reports were monitored, and CRP levels decreased so quickly. We treated, as I said, almost around 1600 patients in these 2 waves, and seeing those follow up, the CRP levels and other symptoms, they improved so quickly with Virafin. And we also gave for some patients
Dr. Nitin, we cannot hear you. Like, maybe I request chair to.
At the moment, we cannot see, we cannot hear the Dr. Nitin Akra. Probably mute, or is the internet connection is something wrong.
Yeah, I think his internet connection could be an issue. I think he's not able to. Okay. So I'll.
Are we?
Okay, so with the permission of the chair, may we proceed for the polling?
Yes, you can. Please do the poll at the moment while waiting again, Dr. Nitin.
Thank you.
So all the participants, so please participate in the pool. We have a pool for the use of in the interferon Virafin. So please participate in the pool, please.
Thank you, Professor Coco. So I will request the administrator to please start the polling. The audience, please note that you will have 10 to 15 seconds to answer each question. Okay, let us go to the second question, please. Admin, may we have the second poll? Let us have the next polling question. Yes, may we have the next question, please? Okay, thank you. Any last question for the polling? This question, you have to type your answer in the box. Next question, please.
Oh, this is the end of poll.
Thank you very much.
All right.
So I will request the chair, Professor Coco, to begin the Q&A session.
All right. Okay. Thank you. So when we looking at the questions, we can see a lot of questions. So my first question is, which one do you prefer, ivermectin or favipiravir for mind cases? Might be going to the Dr. Deepak. So Dr. Deepak, please. Dr. Deepak, are you there?
I didn't get the question. Can you repeat the question, please, Professor Coco?
Sure. Which one do you prefer, ivermectin or favipiravir for mind cases? That is the question.
So which treatment
I
would
think
you
there's a
prefer
lot of
for mild?
disturbance.
So which treatment would you prefer for mild cases? Is it ivermectin or favipiravir?
Yes. Hello? Dr. Deepak, are you there?
Can you hear me, sir?
Now I can hear
Can
us.
you hear me now?
Yep, yep. Hello? We not hear you now.
Professor Coco, maybe you can take another question. We can come back to Dr. Deepak Talwar later.
Okay. So another question maybe, what are the expected side effects if a patient is accidentally given a higher dose of? Dr. Kevin or Dr. Sanjay?
Yeah. So I think Professor Coco, I think in between I lost you. So will you please repeat the question?
Dr. Diwa, are you there?
Kevin here, sir.
Kevin, all right. Now, you can see what are the expected side effects if a patient is accidentally given a higher dose of pegylated interferon alpha 2b.
Okay. So in this our study, we have used 1 microgram per kg dose. And as per the package insert, the dose of the Virafin alpha 2B in chronic hepatitis B and C, that is on higher side. That is 2 microgram per kg, and that we are giving every week. So somehow accidentally, if you are giving the higher dose of the pegylated, I don't think there will be any kind of the severe or the serious side effects.
Right. Thank you. So, as you are being answering another question is how should pegylated interferon be injected? What is the dose?
Uh, it's a injections. We are giving subcutaneous injection.
Hello?
Yes. The 1 microgram per kg dose that is given by the injection with the 26 gauge needle, the subcutaneously.
Thank you. Thank you. Another question is, Dr. Kevin, what would be the ideal to administer Virafin after the onset of symptoms?
So yes, so normally the antiviral therapy earlier is better to prevent the progression of the disease. So
Yeah.
if you give the therapy early, so the patient we can prevent the viral replication. And as we know, the RT-PCR value, we have a very good result with the antiviral activity of the drug, and we have a similar profile in phase 2 as well as phase 3. So the early the therapy, that is the better.
Okay. So you mean, sir, Ali the one is a better one, right?
Yeah.
So another one where I think is going to the Kemeni side. How can we get interferon in Myanmar? What's the cause? Is it more effective in without remdesivir group?
I think that is a question for Mr. Ram.
Yeah. So for the Dr. Kevin, is it more effective in without remdesivir group? So I think so you already given the, so they can repeat the question, right?
Right. So, but we may tell because the sample size perspective, we may not conclude ki if without remdesivir or the with remdesivir that is more effective. But yes, we have a data of the without remdesivir, and without remdesivir there is no change into the efficacy.
Right. Thank you. So most of the question will go to the Dr. Tilwa.
So
And
Dr. Talwar is back. We can take up questions for Dr. Talwar now.
now, is he now?
Yes.
All right. So Dr. Tilwa, can you hear me?
Dr. Tilwar has to switch on the mic and the camera.
Dr. Tilwa, can you hear me? He's not, sorry. So Mr. Ran, How can we
So-
get interferon in Myanmar? What is the cause?
Yes, thank you, sir. Thank you so much. Seshadri, I would request you to present my slides very quickly. I will brief about it so that it gives me an opportunity to summarize what speakers has presented while we are waiting for Dr. Deepak Thakur. Seshadri, can I have the slides? Seshadri? Okay, I will share from my end then. Okay. So thank you very much, first of all, for being here. So Zydus, always, we have presented first to Myanmar, and as you know, most of you remember, we are the company first to introduce remdesivir to Myanmar. We donated in the first COVID wave, and we are happy and proud about that. And this is the second time, we are bringing something new for the fight against COVID management. So intelligent start ensures better outcomes in COVID-19. That's what we have heard from Dr. Deepak Talwar and other presenters. So presenting an intelligent solution with Virafin, as Dr. Sanjeev expressed in his slides, that type 1 interferon deficiency is a hallmark of severe COVID-19, and it may be avoided through early interferon administration, because we have seen that very clearly that COVID virus has got a trick to really not allowing the body, the host, to produce the interferon in some of the patients. And these patients, they develop to severe critical COVID as compared to patient who has efficient function and activity. So when we look at the Virafin, the scheme of possible COVID-19 cases with and without early interferon therapy, and we have seen that in patients where the interferon levels are low, and the patient, the cytokine level reached high during day 14 and day 28, and as compared to patient who had interferon therapy administered at an early stage, we could really control the cytokine levels, and the body's innate immune response could able to overcome the disease. So this is another interesting summary I'm picking up from Dr. Sanjeev, because there are some questions also asked, like, how do we face with remdesivir and favipiravir? So Virafin has got a significantly more potent as compared to other antivirals. It requires only about 0.3 nanogram IC50. So in fact, it is 10 million times more potent than remdesivir. And at a recommended dosage of 1 microgram per kilogram body weight, as you can see that is almost double the Cmax level achieved with Virafin. And not only that, the pegylated interferon ensures that even with single dose, the level of the drug is maintained for more than 200 hours, almost 7 days. So you can effectively manage the production of interferon and arresting the replication of the virus, facilitating the innate immune response by the host. So that's a hallmark of Virafin. So this is another very important slide, I would say.
Mr. Ram, we can see your slide and we can hear you. We cannot see your slide and we cannot hear you as well. Hello, Mr. Ran. We cannot hear you. We cannot see your slide as well. You are muted.
So, since Ram sir has some internet issues and technical challenge,
Right.
let me
We can see their pool.
now take the opportunity to invite Dr. Saw Nain Wei, Deputy Managing Director of Pacific AA, who are our partners in the distribution of Virafin. Dr. Saw Nain Wei, please. Dr. Saw, you are on mute. Please kindly unmute yourself. Yes.
Hello, good afternoon.
Good afternoon.
Uh, I'm very honored and delightful to be here with you today, our honorable speaker, professor and guests. I'm very honored and delightful to be here with you today. We, A Medical Products Limited, really proud to distribute Virafin for the people of Myanmar together with the Zydus Cadila. Virafin will arrive in Myanmar tomorrow with a flight, and it is great opportunity for our medical profession and the people. We are planning to sell with 100,000 shots per vial, and I hope it is a very reasonable price and affordable price for our people. It is a cold chain product, and it is important to store at 2 to 8 degrees Celsius. And we are going to provide urgent delivery and commit to deliver within 24 hours for the cities that we have offices. Thank you so much for your support, and we will try our best to serve the medical professionals and people of Myanmar. Thank you so much.
Professor Coco, your mic is muted.
Thank you, Dr. Saw. And we are very happy to hear that the Virafin is coming tomorrow with the landing. And then you are selling with a very reasonable price. It's a one shot, just one shot for one leg. This really reasonable price and very useful for all the doctors who are treating the patient at home. So because remdesivir, as you know, we need the IV line, and then we go for 6 days, and then we need a 6 vial. It costs a lot. So even though the favipiravir is also a good one, and it also costs a lot as well. So that is very good news for us, right? Thank you so much.
Thank you so much, Professor.
Thank you, Dr. So. And Professor, with your permission, now I'm back.
Yeah.
There was some technical snag.
Please do.
So, with your permission, so presenting an intelligent solution for COVID management from Zydus, Virafin. And as you know, as Dr. Sanjeev was making a mention in his discussions, that COVID has got a trick of making the host not producing sufficient interferons. So type 1 interferon deficiency is a hallmark of severe COVID-19, and it may be avoided through early interferon administration. As you can see, and patient with efficient interferon production and activity, the disease become mild to moderate, while a patient with impaired interferon function, interferon production, and the patient tend to lead to severe or critical COVID. And this is another slide, which talks about scheme of possible severe COVID-19 phase without and with early interferon therapy. So when we have seen that, with patient with low interferon level, the certain levels, goes up, and they get up, some of them into severe disease. And when early interferon therapy was administered, and we could get into only moderate disease and early recovery. So, as Dr. Sanjeev was making a mention about the potency of antivirals, Virafin, as you can see, The EC50, IC50 is about only 0.3 nanograms per milliliter. And so this is almost 10 million times more potent than remdesivir. And Virafin achieves, like, with a standard recommended dose of 1 microgram per kilogram body weight. As you can see that, significant levels are achieved. And not only that, with single shot Virafin, the drug levels are maintained for more than 200 hours, almost for 7 days. So that's why, the patient requires just a single shot. And this slide talks about very important slide. This talks about the timing of interferon, as Dr. Deepak Talwar was making a mention. Is it about the drug or the timing? It is both. So with interferon, when we start early, we can see that there is a reduction in the viral load. And when there is a delayed interferon, we can see that the viral load is very significant. And when there is an interferon deficiency, the patient, we can see that the viral load is very, very high. So early treatment with Virafin will definitely ensure a significant reduction of the viral load, so thereby facilitating an early recovery from the disease. So this is the phase 3 trial, which I quote from Dr. Kevin. So higher proportion of patients in Virafin arm shown to improvement on day 8 versus reference arm, and RT-PCR was negative by day 7, and there is a reduction, hours of supplemental oxygen required, and time to resolution of sign and symptom was significantly less in Virafin arm. And I'm coming to the most important slide. I think this is going to be very, very practical, because the dosage is 1 mcg per kilogram body weight per dose, along with recommended SOC, and route of administration is subcutaneous, and method of preparation is very important, and it's simple, because in Virafin you will have Virafin lyophilized powder. We are going to provide water for injection 1 ml, and there is a 20 gauge needle, there is a 26 gauge needle, and there is a disposable syringe. So Sheshadri, you can mute others. I'm getting echo. So the first and foremost, we have to draw points 7 ml, push into the vial, shake it, mild shaking, rolling, welcome a clear solution, and take it into now 26 large inch and administer as a subcutaneous injection. So 0.5 ml equivalent to about 80 mcg is a dosage, and storage recommendation is 2 degree to 8 degree, and it should not be frozen. And you can, and this product comes with the product information catalog, so you can refer for further information. And price, we have kept very reasonable as compared to other therapies which are available. When we compare, it is very reasonable. It will cost, as Dr. Sonone said, it will cost about 1 lakh chak. So thank you very much. And I also request now Professor Coco to take over from my side.
Thank you. Because we cannot contact with Dr. Tipat Dawa, and there are a lot of questions stay there. But with the time limit, I would like to wrap up the symposium. I would like to close the symposium. So thanks to all the speakers, and first of all, I would like to thanks to the Dr. Tipat Dawa with sharing his experience and Indian experience, and that will be a lot learned for us. He is really talking about in the viral phase, don't give the dexamethasone. He mentioned about all the antiviral drugs, and then we can learn one new one. This interferon. In spite of we are using Brava in remdesivir in our country, we are now new ones is coming up with the interferon. That's a really promising one. And then he mentioned that in the other stage, the dexamethasone is really important, and you can use the midnight as well. and he mentioned about tocilizumab and the paracetamol, everything. So thanks to the Dr. Gibak. Also, Dr. Sanjay Kumar is talking about the science behind the giving the Virafin for us. So interferon deficiency is really important in the COVID-19. So why we using the interferon for that? So thanks to the Dr. Sanjay Kumar as well. And the Dr. Kevin also talking about all the experimental findings of the pegylated interferon when we use it early and without remdesivir, with remdesivir, steroid, without steroid, there were less side effect and then more bara clearance and then and more negative and the PCR in the 7 days. So these are the things that we have to learn. So thanks to the other speaker and thank you to the Mr. Rai and the Zydus team for bringing us a new weapon for fighting against the COVID-19 in Myanmar as well. Thanks to you all. And then I would like to hand over to the Dr. Hadik for continuation. Thank you.
Thank you, Professor Coco. And with that, we would like to wrap up this symposium with a few words and vote of thanks from Mr. Ram, our country head for Zydus Myanmar. Over to you, Ram sir.
Thank you, Dr. Hadik. Thank you, Chair, Professor Coco. I must thank, Chair. This symposium was arranged in a very short notice. I think this is the shortest symposium time we have made within say about 5 days. We could arrange a symposium and organize it. So thank you very much for the Chair, Professor Coco, for your guidance. And I must acknowledge Dr. Deepak Talwar. I've known him for more than 20 years. And suddenly I called him, sir, no, this is what we want. We would like to hear your experience about treating COVID, Indian experience. And he was too glad immediately, because he's a very, very busy practitioner. And he immediately gave the time for us. Unfortunately, we could not, I think he is back. I think Dr. Talwar. Dr. Talwar, you are back. Maybe, after I post it, I would still, we will use your presence here to answer some of the questions which is to be answered. And I thank Dr. Sanjeev, SBR torchbearer for the Research Center of ZRC. Thank you very much, sir, for your beautiful presentation. And Dr. Kevin, thank you so much. I think the kind of work we have done for Virafin is cannot be explained in words. Because we have such kind of work we have seen only in multinational things. The kind of detailed work we have done is something amazing, and it gives a lot of confidence to each one of us that this Virafin is one of the safest drug, effective drug in the management of COVID when it is going to be used early. So thank you so much, and I also thank our partner. Dr. Sonuni, Deputy Managing Director for Pacific AA. Thank you very much, Dr. So. And in fact, the drug will be available landing tomorrow afternoon. And she has promised us, with all support, that all possible help she will do to see that the drug is available at an earliest date. Maybe 19th or 20th, it should be available for Myanmar. It will be available. And thank you, Dr. Hardik, for very good coordination and your scientific inputs for conducting this symposia to this extent. Thank you so much. I also thank Sheshadri, who's at the back end. This is a platform we are using for the first time. Thank you, Shesh, for arranging this again. And I take this opportunity to thank our top management, senior management. Sri Ganesh Naik sir is our COO and permanent director of Zydus Group. And I take the opportunity to thank our president, Dr. Amrut Naik, who is also present in this symposia. See, he wanted to attend this symposia as an attendee. He could have very well come as a speaker or a presenter. He said, I would like to be an attendee. And thank you so much, sir, for sparing your time and giving all kind of a support for making Virafin available here in Myanmar in shortest time. And I also personally thank Mr. Manjul Mishra. He's our Senior Vice President for SB1 for all help in making Virafin available for Myanmar. And I also thank Mr. Zumokain. He's the one who really helped in coordination with FDA And I thank our FDA and MOHS for making this drug available in a very shortest time. And I thank each one of you, the participant, for an audience. We recorded more than about 400 participants in a busy day. Thank you so much for sparing your time. And thank you so much. And we can use, I think Dr. Deepak Talwar is back. So we can use his expertise in answering some of the questions. Thank you so much. Yeah, over to you, Prof. You're mute, sir. Your mic is mute.
Yeah. I'm sorry we lost you. And there are a lot of questions coming up to you. Can you hear me?
Yeah, I can hear you.
Right. One question is, which one do you prefer? Is it ivermectin or is it favipiravir in mild cases? Can you hear me? Which one?
Yeah, intermittently. I can't hear.
Which one do you prefer? Is it ivermectin or is it favipiravir in mild cases?
Favipiravir in mild cases.
Favipiravir. Thank you. Another question is, can you use ivermectin for a prophylaxis for the HCP people?
So there are studies from Peru which shows that it was effective when it was given post-exposure prophylaxis and prophylaxis up to 2 months. So we used it, but then the question is how long for the healthcare workers to use it, because this will be indefinite. So that's the reason now it is only the post-exposure prophylaxis. Without exposure, it is becoming too long and it cannot be sustained for that long. So that's why it's only for a immediate post-exposure, and it's more used by the family members.
Thank you. And so another one is, in COVID-19 confirmed case, do you prefer remdesivir or favipiravir or ivermectin or pegylated interferon? This is
In moderate COVID,
they cannot mention, just the confirmed case. In
I couldn't get the
COVID-19
question.
confirmed case, do you prefer which one? Which antiviral agent? Remdi or Favi or ivermectin or pegylated interferon? You cannot. That question did not mention
It
any.
depends upon the severity of the disease and where we have choice, and we will use remdesivir. But if the patient is not in the hospital, and then we have to use either favipiravir or pegylated interferon.
Thank you so much. Thank you. And then and any did another one is, did the patient who were prescribed on basilitinib show increased incidence of thrombosis? Basilitinib is said to increase the risk of arterial thrombosis. Basilitinib with the
Not
thrombosis.
in particular, because they are all on anticoagulants also. Yeah, so I said not in particular because all of them are on anticoagulants, but thrombosis in the pulmonary circulation is being observed, and that is a routine if the patient remains hypoxic. After 14 days, we do angiography to look for pulmonary artery emboli.
Thank you so much. Another one is,
but not ischemia.
Yeah, what is the best time for giving the basilitinib, in the early phase or any phase? Or this is the question. What is the best time to give basilitinib?
So it is being used at the moment in the moderate COVID, as soon as the treatment started, which is with steroids or with remdesivir.
With the remdesivir. Thank you so much. Another question is, do you suggest to give vaccination for liver transplant patient? Vaccination for liver transplant patient. and which type of vaccination?
Yeah, we are recommending vaccine. So now the data is there for the organ transplant patients that all of them can be vaccinated either with Pfizer, Moderna, or the Oxford vaccine. Only thing is that even after the 2 dosages, they may not have sufficient antibodies. So in that case, we are now looking forward for a booster vaccine for them if they don't have a neutralizing antibodies.
Thank you. One thing is, any side effect of thrombocytopenia or anemia after Virafin?
I have not used Virafin personally in large number of patients, so won't be able to comment. I think anybody from the team Virafin will be able to answer that.
Thank you.
Dr.
Thank you.
Kevin can
Dr.
take
Kevin?
that question.
Yes.
Any,
So
any?
yeah, in our study we had analysis of the hematology parameters. There was no any trend with the pegylated.
Thank you. I think sir, I almost covered most of the questions. So thank you so much to all the speakers for sharing your time.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you,
Bye.
Thank
everyone.
you. We come to an end of this symposium. Thank you all for joining. Thank you all the speakers, and please stay safe.
Thank you. Bye-bye.
Thank you,
Bye-bye.
Hardik.
Bye-bye.